So I'm going to talk about today, I'm going to show you some T1D exchange data. First, I'll start with telling you what that is. And thank everyone for not drinking so much last night that you were able to get here at 9:30 this morning. So I'm going to start off giving a background on T1D exchange and the clinic registry, so you have a feel for what that is. And then I'll go over some of our top findings with regard to technology use, glycemic control, and acute complications. And then also as part of the T1D exchange, we did a number of studies outside of a registry, which was just data collection. We did a number of studies, and I'm just going to go through a few highlights of those, and then I'll summarize. So some of you may have heard of Leona Hemsley. She passed away in 2007 and left all of her money to her dog. But eventually that money ended up in a trust, and it was referred to as the Hemsley Charitable Trust. And one of the trustees for that trust was David Panzier. And shortly after he became a trustee, his oldest daughter, Morgan, developed type 1. And so he made it his mission and passion to figure out what was missing in type 1 research and focus the Hemsley Trust efforts onto filling that gap. So he hired a number of people to kind of help him figure this out. They literally traveled the US just talking to specialists in type 1, trying to figure out what was the current status, what was going on with research, what was missing, what needed to be done. One of the people they talked to was my boss, our executive director of the Jabe Center. His name is Roy Beck. And after meeting with him several times, they came up with the idea that to form a clinic network of endocrinology clinics throughout the US, and one of the first initiatives of that network to develop a type 1 diabetes registry. So just to enroll people into a registry all over the US and figure out, outside of a clinical trial, how are people really doing? How are they managing their diabetes? So we hoped initially to attract 25 diabetes centers. We weren't sure how interested people were going to be in this. It turned out there was a huge amount of interest. 57 sites were selected initially. We ended up adding 24 more. This was all going on in the beginning of 2010. In that summer, we developed the data collection. We enrolled our first patient in September of 2010. For about 2 years, we had what we call our core enrollment, where we got about 25,000 patients with type 1 diabetes, and then ended up adding an additional 10,000 between 2012 and 2015. So about 35,000 total that were part of the clinic registry. So as far as clinics go, we ended up 18 adult clinics, so 18 endocrinology clinics that saw only adult patients, 38 that saw pediatric, and 25 that were a mix, so they saw both pediatric and adults. And then the majority of them were institution-based, meaning that they were part of either a hospital or a university, but we did have some that were private practices or managed care. And this is where all the clinics were. So we wanted to get one in every state. We came close, not quite, but pretty good representation throughout the US. So what happened? So we wanted to just collect data as part of this registry. Participants signed a consent form, and then we ended up with about 2/3 of the data that we collected coming from a patient questionnaire. And that was an involved dynamic where we realize that there's not a lot of data on how people are managing their diabetes that ends up in the medical record. That really, to learn about what's going on in the day-to-day, we needed to ask people who are living with it. So we ended up with 2/3 coming from the questionnaire, 1/3 that came from the medical record, and then we did annual updates from the clinic medical record and also emailed questionnaires to patients from 2013 to 2018. So what do we collect? Some demographics, history of diabetes diagnosis, insulin management, glucose monitoring, and some general stuff. We also repeated some of that with the clinic data, but then got other medications, medical conditions, labs, et cetera. We've had one of our more important papers published in Diabetes Care, and I'm not just saying this because I was the first author, provided a glimpse into how individuals of all ages were managing their diabetes. And that was published in 2015, and we've recently updated that data, which was published earlier this year. I'm happy to send these papers to anyone who is interested. So before I get into what I hope is interesting data, I wanted to give an overview of the age and diabetes duration ranges for people who are part of the registry. I guess out of curiosity, is anyone in the room a T1D exchange registry participant? There's one, couple. Or maybe you were, but you don't remember that you were. All right, maybe like about 10, I think, hands I see, cool. So you can see we have a pretty broad range of ages. Decent amount less than 6, and then also, in the thousands for all the other age groups as well. And for duration of T1D, we also have a pretty broad range. So people who have had diabetes for a very short time, and then also we have over 1,500 who had diabetes for 40 years or more. Okay, so some top findings with regard to technology use. So these data were collected between 2016 and 2018, and there's about the total number in these slides I'm going to show you is around 23,000. So first finding, across all age groups, over half of registry participants are using an insulin pump. So this 2010, 2012 is kind of our initial enrollment period, and then we compare that to the more recent data look that we did, which was between 2016 and 2018. So throughout some of these slides, I'm going to show both those years. So you can see it's between 50 and 60% in 2010, 2012, and that increased slightly when we did a relook at the data. But across all ages, about 2/3 are using an insulin pump. So one caveat to keep in mind regarding the T1D exchange registry data is that it's not a population-based registry, meaning obviously not everyone in the US with type 1 is enrolled into this registry. And these data are from patients that are receiving care at many of the top endocrinology clinics in the US. So it's likely that the actual penetration of technology in the country is less than what we're seeing here in these data. Next finding, so CGM use has increased substantially, with the largest increase occurring in youth less than 6 years of age. So when we looked at initially enrolled, there's 7% overall participants were using CGM, and this is again around 2011 for the most part. You can see about 15% of adults were using CGM and around 3-4% of children. Then when we looked at this again, it skyrocketed. So in those less than 6, it went up to 52%. I think that's in large part due to like the Dexcom share feature, where parents could actually have their phone and be sleeping at night in their bedroom and get alarms on their phone for kids. But it jumped in all age groups. And this is data from 2018. I would imagine the 2019 data, if we had it, would look even higher. So you can see here, it just highlights the sharp increase in CGM use that has occurred over the past 5 or so years. Again, this is through 2017. You can see it really started kind of kicking off in 2014 and then just incrementally increased over time. Next finding, so use of CGM is more common among participants using an insulin pump, but is also used by a substantial number of adults with T1D who use injections for insulin delivery. So this is the percentage of participants using CGM. In the dark blue, it's the people who are using a pump, and then their percent using CGM. So you can see kind of across the ages, it ranges from about 30 to close to 70% in the young kids. But there's still a decent amount using injections. And I think it's important to recognize that some people with type 1 may never want to use a pump. They might want a CGM with injections. And this is particularly true for young kids where real estate on their skin is an issue, and then also sometimes for older adults where real estate becomes an issue because of tissue scarring and that kind of sort. So you can see the percentages there. Okay, so I'm going to move on to looking at some of our top findings with regard to A1C and glycemic control. So first, most adults and children with T1D do not achieve A1C goals set by ADA. So Aaron actually showed this slide yesterday in the talk that he gave. We were hoping that with increase in technology, that when we looked at our follow-up data, we would also see an increase in the proportion of participants who were meeting A1C goals. But we didn't. And I think these data just solidify how hard it is to achieve these goals, where you can see only across the board, adults, about 30% of adults are achieving the ADA goal of an A1C less than 7%. Adolescents and young adults with T1D have higher A1C levels than younger or older individuals with type 1. So this slide has been shown a number of times. If you go to a research conference, you may see this slide. And we decided to plot A1C over ages, over the course of the lifespan, to get an idea of the pattern. And you can see there's this peak in A1C in adolescence where the average teenager starting college has an A1C above 9%. And it really doesn't level out until they hit about age 30, and then it kind of stays pretty steady. And this increase in A1C isn't unique to the US. So this is showing data from several European registries, and the T1D exchange included. We're in this greenish color there. You can see the US has kind of higher A1Cs overall than a lot of European countries, but in general, you're still seeing this rise in A1C in adolescence, regardless of, kind of what country you're in or where you're starting. So they might have figured out how to have lower A1Cs, but they haven't figured out how to teach adolescents to be normal people. So next finding, CGM users have a lower average A1C regardless of insulin delivery method or level of household income. So this is looking at that same curve of A1C across the lifespan, but stratifying it by CGM use. So the red line is people who are not using a CGM, and the blue line is CGM users. And you can see overall, despite the age, there's about at least a 1% difference in A1C. So again, you still see that peak in adolescence, but it's not near what it is in those not using A1C. It's 8.3% about in CGM users in adolescents compared with 9.5% on average in those not using a CGM. And then when we get into an adulthood, it's 7.4% in those using a CGM compared with around 8.3% in those not using a CGM. So when we look at this in technology use groups, so the red line is someone who doesn't have a pump or a CGM, so no technology. And the green line is someone who's using a pump but doesn't have a CGM. And you can see that it improves your A1C. But then if you add on the CGM, really, whether you're using injections, MDI, multiple daily injections, or a pump, pretty much have about the same average A1C across the lifespan, particularly in adults. And this holds true with regard to socioeconomic status. So here I'm using income as a representation of socioeconomic status. But you can see A1C is lower with CGM use across all income levels as well.
Do you have data with a lower cutoff, a lower income cutoff, like 30 grand?
We do, yeah, but it's as you might expect, the number of CGM users when you look that low is pretty low. You can't even really plot it. Yeah. So looking at now acute complication, so severe hypoglycemia and DKA. These data are actually from participant reported, because that's where we found we got the best accuracy of when these events occurred. So it's around 11,000 that completed this questionnaire. First finding, severe hypoglycemia resulting in seizure or loss of consciousness occurs more commonly in adults with long-standing type 1 than has been appreciated. So this graph is showing severe hypoglycemia in the past 3 months, and it's defined as an event that involved either seizure or loss of consciousness. So we wanted to make it a kind of a hard, clear-cut definition. So it's not just someone like going really low and they drank juice and then we're okay. This is an event that involved seizure or loss of consciousness. And you can see that in adults, this is, I mean, over 7% are saying that they've had an event like of this severity in the past 3 months. And it's really an aspect of duration. So participants with 30 years or more duration have almost double the risk of having a severe hypoglycemic event involving seizure or loss of consciousness in the past 3 months than those with less than 30 years of duration. I mean, we actually did a study looking at that. It was hypoglycemic unawareness. I mean, as I think many of you probably would guess.
But these results all reflect people who were using no technology for most of their diabetes life.
Yeah, yeah. In both children and adults, the frequency of severe hypoglycemia is higher for both those with really low A1Cs, but also those with really high A1Cs. And this is in contrast to the findings of the DCCT trial that took place in the 1980s. So the DCT found that you could lower your diabetes complications with lower A1C. But the lower your A1C, the higher the risk of severe hypoglycemia, where people with high A1Cs had very low risk of severe hypoglycemia. And what we've seen in the T1D exchange registry data is different. So it's almost like a U shape. So participants with A1C between 6 and 7% have less severe hypo than those with an A1C above 10. So you can see there's kind of an increased risk if your A1C is less than 6, but there's also an increased risk if your A1C is above 10, 11%. And that has to do with a lot of like glycemic variability, kind of going up and down a lot. The frequency of severe hypoglycemia is less common among those using an insulin pump or CGM. So this is showing severe hypo, again, seizure or loss of consciousness in the past 3 months. You can see the percent who reported having an event who were pump users overall was around 5%, and that was almost doubled in those not using a pump, around 9%. And then when we look at this by CGM use, again, less severe hypo events with CGM use. This becomes tricky to look at because a lot of times people start a CGM because they have a history of severe hypoglycemia. So I think we would really see like even a bigger discrepancy if you could kind of tease out the fact that someone has a history, so that's why they got on CGM in the first place. Diabetic ketoacidosis occurs more frequently in youth and young adults and among those with high A1C. Maybe not too surprising, but what was surprising is that we saw, and these are DKA events that involved overnight hospitalization in the past 3 months. Over 5% of youth are reporting having had a DKA event in the past 3 months that involved overnight hospitalization. And if you have an A1C of above 11, 17% are reporting having a DKA event in the past 3 months that involved overnight hospitalization. So DKA occurs less frequently in insulin pump users than injection users in youth and young adults, but about a similar rate in adults. However, with CGM use, we see much less DKA than those not using CGM. So this first slide here is looking at DKA by pump use. Again, overall, less in pump users than non-pump users, about half. When we look at it, it's much more prominent in kids, and then when you look at it in adults, it's a little less, but not that much different. We're looking at CGM use. Again, it's about half, cut in half. If you're using a CGM, your risk of DKA is about half of those who are not using a CGM. So I'm going to move on to highlighting some highlights from studies that we've done outside of the general registry. So we did ask when we enrolled people in the registry, we asked if they would be willing to be contacted via email about studies that they might be eligible for, that we could kind of assess that they were eligible from the data that they had provided for the registry. And 75% of participants agreed to do this. And so we were able to do a number of clinical trials pretty quickly because we could kind of look who was eligible, email them, give them their clinic's contact information, and it just sped along the process of getting research done. So we did a number of studies, about 12 studies, most of which were funded by the Hemsley Charitable Trust. However, a few of them were funded by JDRF. So the first study I'm going to highlight we called Replace BG. So this study was done prior to the FDA approval for the Dexcom CGM to be used to dose insulin without performing a finger stick. That happened in late 2017. The objective was to see if people who were assigned to use CGM, they were already CGM users, we took all CGM users, and then randomly assigned them to either use a CGM to dose insulin and not perform a finger stick to dose their insulin, or to use the CGM but also use their blood glucose meter and do a finger stick to make their insulin dose decisions. And the objective was to see if it was safe, basically, to use your CGM to dose insulin as opposed to a meter. And so we found out it was. So this first line here, the CGM only group, so they're the ones who are using CGM to dose insulin. They started out at the beginning of the study with 63% time in range. They used CGM to dose insulin for 6 months, and they ended up with the exact same percentage of time in range. And their percent of hypoglycemia was also the same. I'm not showing that here, but. And then for the group that used CGM but also did BGM, they again started out at 65% of time in range. That's the difference there is just coincidence, and stayed the same. So we found that it was safe to use CGM to dose insulin, and these data were provided to the FDA to help them make the decision to approve the Dexcom G5 at the time for use not adjunctively or to be able to dose insulin. So the next study I'm going to highlight is on racial differences in glucose concentrations in hemoglobin A1C. So there was some question as to whether or not African Americans had a different glycation process than non-Hispanic whites, where they the way that hemoglobin is glycated raises their A1C. So for any given glucose, so a glucose of 200 may equal a different A1C in an African American than white non-Hispanic. So we wanted to assess if that was the case. And part of this was driven by registry data. So this is from the clinic registry, where we saw extremely higher A1Cs in African Americans compared with whites, where overall the average A1C in an African American was 9.7% compared with 8.3% in a white non-Hispanic. And so we conducted this study. They were blinded CGM for 3 months, the Abbott Libre. It was replaced every 2 weeks. And it ended up that there is some sort of glycation difference between African Americans and white non-Hispanics. Not enough to explain the large difference that we're seeing in A1C, but enough where there is a difference. So for an African American with an A1C of 8%, that would be equivalent to an A1C of 7.7% for someone who is non-Hispanic white. So we also use the same data to show that A1C is really an imperfect measure. For any given person, their relationship between their mean glucose and their A1C is different than what it might be for someone else, and also different depending on what your glucose level is. So it's really, if someone might have a glucose of 200 and have an A1C that's 8%, and someone else might have a glucose of 200 and have an A1C that's 7.5 or 8.5, and it varies. It's not a direct, perfect relationship. So the next study I'm going to talk about is looking at mini-dose glucagon as a novel approach to prevent exercise-induced hypoglycemia. We kind of touched on this yesterday in the exercise session. And so there's 4 groups in this clinical trial. The control group did nothing, so they exercised. Everyone had the same meal the night before, the same meal, and they titrated their insulin, the same meal the morning, so they're all starting around the same glucose level. There was a control group that did nothing. They all did the same exercise. It was, and the control group did nothing. The basal group had a reduction in their basal rate to prepare for the exercise. And then there was a glucose tab group that took glucose tabs to help their glucose raise back up, and then a mini dose glucagon group, which means they just gave it's a very small dose of glucagon, which is made by a company called Xeris. So you can see here kind of the percentages of going low after exercise were higher in the control and the basal reduction groups than in those who took glucose tabs or mini dose glucagon. And then on the flip side, the percentage who ended up also going high later, obviously was lower in the control and basal reduction groups. But what's interesting here is that the glucose tabs kind of raise glucose a lot higher than taking just a mini dose of glucagon. So with only one person getting their glucose, having a rise in the glucose above 250, compared with 5 people who took the glucose tabs.
How many glucose tabs?
I don't know, actually. I don't know. It was a standard amount, same people, but I actually don't know that offhand.
The conclusion from that study on the previous slide is that, are we to conclude from that probably the preferable way to do it is with glucagon?
I think mini dose glucagon is an option. I don't know if I can say a conclusion, but yeah, we gotta keep going, sorry. So for intranasal, the next study I want to talk about is intranasal glucagon. We've touched on this. This is a glucagon that is sprayed up into the nasal cavity instead of injecting it. We did this study in both adults and kids and showed that it works. It works the same as glucagon and works really well. And again, both children and adults. This was recently, we used the data from the study to get FDA approval. Lilly actually ended up buying the small company that originally had this product that was Leucemia. It is now FDA approved, and it's called Baqsimi? I don't even know how to pronounce it. Something like that. Okay, so quick summary. We've had a lot of publications that have come out of these data. It's been a really great, productive effort. 162 presentations from 2010 to 2019, 75 manuscripts that we've published, and a number that we're still working on. It's really served, the registry data has really served to establish the state of type 1 management and outcomes in the US and illustrates how challenging living with type 1 really is. We've used these data to help support blood glucose meter coverage for insurers. There was, at one time, the state of Washington was saying that kids needed to only check their blood glucose 3 times, and we were able to use this registry data to show, that's not true. They need to do it a lot more often. We've also helped change ADA guidelines for A1C goals from pediatric patients. Again, the FDA decision to expand CGM labeling to include, to not have to do finger sticks for dosing insulin, and also to provide data for CGM to be covered by Medicare. We've collaborated with registries around the world, which has been a really great experience to learn kind of differences in practices. And I think the legacy is just raising an awareness of type 1, the challenges in day-to-day management for healthcare providers, researchers, companies, insurers, regulators, and also people with and without diabetes. Obviously, I'd like to thank the people who participated, and those of 10 or so of you in the room, and our research teams, and then the Hemsley Trust for funding it.